This is From the Lab Lectures, Story of COVID Origin. We've all seen the headlines. Did the virus really come from a lab in China? I'm a virologist. I've got friends and colleagues on both sides of the debate. I dug deep, I read everything out there, and I collated it into a cohesive, one-stop shop explanation, probably the most comprehensive of anything available anywhere. It's for the people that really want to know. Today is about Dr. Fauci, his emails, his connections, and the observation that started it all, the fear and cleavage site. Dr. Anthony Fauci is obviously going to be a main character in this story. At the start of the COVID-19 pandemic, he was the head of the NIAID, or National Institute for Allergy and Infectious Disease. The NIAID is a funding agency. They fund research conducted all over the world, but they also conduct their own research on their campuses in Maryland and Montana. They're a subset of the NIH, or National Institutes of Health, and that's run by Francis Collins, or at least it was at the time at the start of the pandemic. Why Fauci became the face of the pandemic response instead of, say, the head for the Centers for Disease Control or his own boss, Dr. Collins, is not straightforward, but it's probably because the man has an ability to connect with people, as evidenced by his nomination for Sexiest Man Alive and these prayer candles made in his likeness. On February 1st of 2020, Dr. Fauci sent an email to Jeremy Farrar, head of Wellcome Trust in the UK. And we know this because many of Fauci's emails were released upon Freedom of Information Act requests. CC'd on this particular email was Christian Anderson, who is an evolutionary biologist at Scripps in La Jolla. The email subject is phone call. Just pause if you want to read it, but Fauci basically writes, I just got off the phone with Christian. He told me about his concern with the fear and cleavage site in the COVID virus. We should get a group of evolutionary virologists and coronavirus experts together as soon as possible to determine if his concern is validated. If it is, we Americans should alert the FBI, and you, Jeremy, should alert the MI5 in the UK. So Fauci's up after midnight answering this email. On his to-do list for the next day is, number one, validate the concern with a team of experts. Number two, alert the FBI. And he followed through on the first half of this plan. He and his boss organized a Zoom call with a group of American and international virologists. And from the FOIA emails, we know the topic of discussion was the origin of COVID-19, particularly whether the sequence appeared to be bioengineered, especially the fear and cleavage site. 48 hours after the conclusion of that meeting, there was evidence in Fauci's emails that the first draft of this paper was in existence. It's called The Proximal Origin of SARS-CoV-2. It was written by 4 people on the call: Christian Anderson, Andrew Rambeau, Edie Holmes, and Bob Gary. And then one person who was not on the call, Ian Lipkin. Ian Lipkin also studies cross-species transmissions, and fun fact, he was the scientific advisor for the movie Contagion. The preprint of the paper became available online February 16th, so that's about 2 weeks after the call, and the final version was published in Nature Medicine March 17th of 2020. This paper argues that a natural zoonosis is the most likely origin and that a lab leak of an engineered virus is very unlikely, though they do address the possibility. They say, we offer a perspective on the notable features of the SARS-CoV-2 genome and discuss scenarios by which they could have arisen. Our analyses clearly show that SARS-CoV-2 is not a laboratory construct or purposefully manipulated virus. It's difficult to overstate the impact of this paper. By 2023, it has over 5,000 citations. It has been accessed over 5 million times. It is the seminal paper on the topic. Problem: the scientists on Fauci's call continued to discuss the issue of potential bioengineering over days, and they dwelled on the potential of a lab leak origin. Several email quotes look pretty damning, very much like the scientists were warning Fauci that the virus looked unnatural. Here are some quotes. I am bothered by the fear insight and have a hard time explaining that as an event outside of the lab. These scientists are giving a numerical value on their certainty. They're saying, I'm 70-30, I'm 60-40, I'm 50-50 lab. One person saying the only thing striking them unusual is the fear and cleavage site. One person says it's not consistent with evolutionary theory. And in the very worst case, Bob Gary says, I really can't think of a plausible natural scenario where you insert exactly 4 amino acids, 12 nucleotides that all have to be added at the exact same time to get this function. It doesn't help that between them, Christian Anderson and Bob Gary's labs get over $20 million in grants from the NIH and the NIAID over the next 2 years. This is not an unusual amount of money for big labs working on topics of extreme current interest, and it might be a self-fulfilling prophecy where being the authors on this paper gets them scientific fame and then that gets them grants. But of course, the optics are bad. This is something that critics can definitely pick at. Quotes from Dr. Collins further reveal that he was very eager to squash the lab leak conspiracy theory. He said, "A swift convening of experts and a confidence-inspiring framework is needed, or the voices of conspiracy will quickly dominate, doing great potential harm to science and international harmony." This statement left him extremely vulnerable to criticism that he should have been focused on pursuing truth, weighing all data equally, and letting the evidence speak for itself. He is a scientist, not an international relations expert. Like, what is he doing? As lab leak theory gained steam through 2021 and 2022, Congressional House Republicans agreed that these emails were super concerning. They wrote a scathing letter to the Secretary of Health and Human Services requesting a transcribed interview with Dr. Fauci to explain himself. The scientist clearly had a problem with the furin cleavage site. What is that? The spike protein on the outside of the virus particle is shown here. It's a trimer. Each of these 3 individual spike proteins is 2 subunits, S1 and S2. And the protein needs to be cut or cleaved between those 2 subunits to trigger entry into human cells. Otherwise, it just kind of hangs out on the outside of the cell and doesn't really do that much harm. There are certain series of amino acids that tell human proteases to cut here. Furin is one of a whole variety of those proteases, along with heparin, thrombin, plasmin, etc. Furin has a favorite series of 4 amino acids to cut, and that's called a furin cleavage site. The minimal sequence is arginine, anything, anything, arginine, though it's a lot more efficient if it's arginine, anything, lysine or arginine, arginine. This is what the SARS-CoV-2 genome looks like at this spot. This is an alignment comparing it to its closest bat relatives and to the first SARS. And, yowza, you can see how this 4 amino acid insertion sticks out like a sore thumb and might be alarming. In this box are the amino acids that make up the furin cleavage site. So there's this extra proline in the insertion, then RRA, then using an R that's already there. And it makes a minimal furin cleavage substrate of R, anything, anything, R. Here's an alignment with a lot more sequences. It includes SARS isolates from 2003, many more of the closest related bat coronaviruses, and this piece still looks disconcerting. So the scientists have a problem with the furin cleavage site. You can see why. Where did it come from? How did the virus get this perfect insertion right here when no closely related bat viruses have it? 2 weeks later, they've submitted a paper strongly arguing for natural zoonosis. The letter from House Republicans reads, "It is unclear what, if any, new evidence was presented or if the underlying science changed in that short period of time." And I understand the suspicious activity that brings out this anger and sarcasm, but this is a misunderstanding of how science works. Most science is not experiments. Most science is deciding from a whole Earth's worth of disorganized information what's the right information to lay side by side to synthesize an informed argument. So what changed in that short period of time? They did some thinking about what an engineered virus would and would not look like. And they, to say they got no new information is false. They got a pretty key piece of information. They got coronavirus sequences isolated from an animal called a pangolin. Pangolins are mammals with scales. They are the world's most illegally trafficked animal, accounting for 20% of illegal wildlife trade because people believe that these scales have magical properties and they're an integral part of Chinese natural medicine. The pangolin coronavirus sequence comes from pangolins confiscated in the wildlife trade. So point 1 of 4 argued in the paper is that the pangolin sequence almost perfectly matches the receptor binding domain of SARS-CoV-2, and that is way too big of a coincidence for the virus to be unnatural. The receptor binding domain is the part of the spike protein that's the physical interface with the ACE2 receptor. It's a little over 100 amino acids long, and there are 6 sites that are the most critical for binding. This area of the genome has an enormous amount of genetic diversity, so similarity in the specific spot is a big deal. For reference, 5 of the 6 critical sites are different in SARS-CoV-2 than the original SARS. 5 of the 6 critical sites are also different in the closest known bat relative. But 6 of 6 are identical to this pangolin virus. For perspective, that's a 1 in 64 million by random chance. It seems that there are 2 papers that publish the pangolin coronavirus sequences. The first preprint goes live February 18th and the second on February 20th. But Jeremy Farrar had some connection that must have given him access to it a few days early. He says in the emails, reports coming out overnight that Chinese group have pangolin viruses that are 99% similar. This would be a crucially important finding, and if true, could be the missing link and explain a natural evolutionary link. Colin says that would be very interesting. Does it have the furin cleavage site? It just shows you how hung up on that furin cleavage site they are. But it did not. And Farrar did not get completely accurate, hot off the press info. It turns out the virus is only 90% similar overall. It was the receptor binding domain that was 99% similar. Even so, E.D. Holmes, who had previously said 60-40, said this is enough for him. Personally, with the pangolin virus possessing 6 of 6 key sites in the receptor binding domain, I am in favor of the natural evolution theory. Scientists spent a year or two puzzling how these unlikely hosts could be the missing link or the intermediate host. We know that bats are the ultimate reservoir and have by far the most strains, the most diversity, the most ancient infections, but usually there's an intermediate animal bridging viruses crossing from bats to humans. This is the case with Nipah virus. This is the case not with bats, but with waterfowl to humans in the case of flus like H1N1. It's a stunning coincidence that the pangolin, an animal so heavily involved in the wildlife trade, has such a similar virus. But overall, the strain cannot possibly be the immediate progenitor of COVID-19 because 90% sequence similarity is like 100 years worth of evolution away. Plus, pangolins are solitary. They're a terrible choice of natural host for a respiratory virus, so surely there can't be that many strains of pangolin viruses. These animals almost certainly picked up the infection once they entered the wildlife trade in a van or a warehouse somewhere from the animal in the cage next door. They did not get it roaming around in the forest and then bring it into the wildlife trade. And further confusingly, there were no pangolins in the Huanan market in Wuhan. So it's still not clear exactly how pangolins were involved, if they were involved. I don't want to cover up that these particular sequences later become mired in controversy. Multiple Chinese research groups kept publishing the same data. Now, co-publishing is not unusual, and the people complaining about this issue are the biggest proponents of lab leak hypothesis, but there seemed to be a bit more strangeness to this story, and the China of it all just kind of tarnished the believability. There were also some concerns with PCR contamination, but at the time, the proximal origin authors were acting in good faith. The pangolin coronavirus sequence was really compelling evidence. It was simply too big of a coincidence for SARS-CoV-2 to not have something to do with the wildlife trade. The second point the proximal origins make for SARS-CoV-2 not being bioengineered is that it has the same glycosylation sites as its relations in the bats. Glycans are sugars. We tend to think of proteins as just their amino acid sequence, but they're often covered in sugars. Specific amino acids kind of signal to attach sugar here. And viruses make prolific use of this system to hide from the immune system. A big old protein is very recognizable as non-self, but if it's covered in sugar, it's kind of like an invisibility cloak, and it can kind of hide from the immune system because sugars are the same between a lot of creatures. Where are we going with this? The scientists in the email thread postulate that maybe the virus arose by serial passaging in human cells. Serial passaging means putting the virus on one dish of cells, a few days later collecting the virus that grew out, putting that on a new dish of cells, and repeating that process over and over. This is a way to passively select for mutations that could make the virus better adapted to human cells. And I know it's not intuitive, but allowing evolution to do its thing and pull out the winners after several blind tries is way more effective than a human sitting there trying to design something. But the authors argue that the furin cleavage site could not arise in serial passage in human cells because when viruses are passaged in tissue culture, they are not subject to an immune system. They have a strong tendency to lose those glycosylation sites. But SARS-CoV-2 has all of them, just like its that relatives, therefore not created by passage in tissue culture. Here's a FOIA email from February 8th on the glycans. Quote, the existence of glycans is pretty strong evidence of evolution in the presence of an immune system. The authors do not address, though, what if the furin cleavage site arose by passaging the virus in an animal? Fauci himself asks, would serial passage in an animal in the laboratory give the same result as prolonged adaptation in animals in the wild? This is not my area of expertise, so I do not know. Along a similar line, The Intercept reports a conversation that I could not actually find myself, but it reads, Fauci says, what if it was passaged in ACE2 mice instead? And then Colin says, surely that wouldn't happen in a BSL-2 lab. And then I imagine Farrar takes this drag off of his cigarette and says, Wild West. Yikes. Okay. On the one hand, Fauci brainstorming unnatural explanations of origin is not behavior that's consistent with someone dead set on silencing debate and paying people off. On the other hand, the paper does not address a resolution to this question. It is a hypothetically possible scenario. The reason it is far-fetched is that it's one thing to passage a virus on a new dish of cells every 3 days for 6 months. But mice are difficult to work with. They have to be fed. People have to work on the weekend. They have immune systems. They're expensive. There's no history of Zhang Lixi's lab publishing anything like this. And it's really difficult to imagine what purpose it would serve. But it is hypothetically possible and not mentioned in the paper. The third point the authors make is that while none of the closely related bat coronaviruses have a furin cleavage site, the spike protein is incredibly variable, particularly in that region. There's this concept in molecular biology and evolution called conservation. High conservation means there's a zero tolerance policy for change in sequence. Maybe that part is in the inner core of the protein doing something structurally important, holding it all together, or for some reason it just has some super important job. If there's a mutation here, forget it. The virus dies immediately, so you would never sample it. You would never see it when sequencing viruses. It's outcompeted by all of its normal neighbors. You only see the normal neighbors when you sequence. What you see from sequencing many different viruses is that they all look pretty much the same because there's little mutation here over time. Then there are regions that are a little more loosey-goosey, less conserved. Maybe that part of the protein isn't really doing an important job, so it acquires mutations, even insertions and deletions of multiple amino acids, because changes aren't really that detrimental. They're neutral here. Viruses make mistakes in copying themselves all the time, but here there's no consequence, so they just get carried on. The furin cleavage site is in a pretty variable region when you zoom out to a wide lens of beta coronaviruses, so it's not unbelievable that the virus could acquire that 4 amino acid insertion. Lots of other coronaviruses have a furin cleavage site right there. In fact, possibly the majority of them do. And in pink in this tree are all the different types of coronaviruses that have them. This includes 2 of the 4 human common cold coronaviruses. It includes MERS, coronaviruses in rabbits, camels, dogs, cows, pigs, horses, and rodents. It's just that all the coronaviruses that seem to be most closely related to SARS-CoV-2 don't have one. But it's clear that furin cleavage sites evolved independently multiple times because they were sprinkled throughout the tree. In some lineages, there's furin cleavage sites. In some lineages, there's not. It's kind of like birds and insects both evolved wings. We all intuitively understand that those are independent evolutions, not one winged common ancestor. So evolution solved the need for wings multiple times. Evolution solved the need for a furin cleavage site multiple times. And there's a distortion here. On big evolutionary time, like 10 million years, clearly furin cleavage sites are seen evolving all over the place. But on small evolutionary time, 500 years, it feels way less plausible when you look at it. But you've got to remember that it just feels that way. This clearly happens. The authors then state that bats are massively undersampled, and they predict that a sequence will be identified in the future in nature that looks like the furin cleavage site of SARS-CoV-2. Point number 4 of the paper is that lab work almost always involves a well-characterized backbone or reverse genetic system in technical jargon. It's the trusty kind of base virus sequence that a lab worker could splice new stuff into to test how that new stuff behaves. For example, in the HIV field, nearly all experiments are done in the backbone sequence of just 3 or 4 strains, or what's called viral clones. They've been around since the 1980s. All labs use the same ones, and they just build off of the information from each other's findings. If I'm doing work on my favorite gene, I might mutate my favorite gene, or I might splice in my favorite gene from different viruses, but I keep everything else in my trusty backbone the same. Otherwise, my experiment is hopelessly confounded by the other 90% of the genome also changing. I haven't isolated the trait or the portion of interest if I don't have a common ground in the parts not relevant to my question. When we say from the lab, for almost everyone's theories, the lab in question is Dr. Zheng Lixi's lab at the Wuhan Institute of Virology. Since she discovered that fruit bats are the reservoir of coronaviruses in 2005, she's built her whole career traveling around China, sampling bat blood, saliva, guano from bats in these caves, sequencing those samples, looking for coronaviruses, and most of my entire second episode is going to be devoted to explaining her work. But in short, for now, she wants to know what it is about a spike protein that allows a coronavirus to bind human receptors, replicate in human cells, because the vast majority of coronaviruses can't do this. The vast majority of coronaviruses pose very little risk to humans. Was SARS a once-in-a-millennia deal? Are there other risky viruses waiting in the cave just around the corner? Answering these kind of questions are her research purpose. To the proximal author's point, Zhang Lixi's lab regularly utilizes only 3 viral backbones: WIV1, WIV16, and SHC014. What they do over and over based on publication record and grant proposals is the following workflow. They collect a bunch of bat samples, they extract the RNA, they sequence the spike gene, they synthesize spike proteins alone, and then they test if those bind to ACE2. The winners they then splice into 1 or 2 of their known to work, i.e. known to replicate in human cells, known to make my sick bat coronavirus backbones. So a long-winded way of getting to this point, but the argument here is that if a bioengineered virus escaped from her lab, based on everything that we know about her publications, the most likely thing for it to be is WIV1, WIV16, or SHC014, but just with a novel spike sequence. That's the vast majority of the quote-unquote risky bioengineering that they do. But SARS-CoV-2 looks nothing like this. If SARS-CoV-2 was WIV1 with a spike sequence that had never been published, it would be painfully obvious that that virus came from the lab. But the SARS-CoV-2 sequence is novel the whole way through. In summary, in 2 weeks, the authors put together as compelling of an argument as they could for a natural zoonosis origin, but 3 of their main 4 points have asterisks by them. They don't know it at the time, but the pangolin sequences are going to become suspicious. They don't address why the furin's cleavage site could not have arisen in animal passage, even though that was something that Fauci and Collins were specifically concerned about. And then a lab variant escape would most likely involve a reverse genetic system. Yes, but not always, not necessarily. Is there wrongdoing amongst Fauci's group of scientists? One view is the I said what I said view from, like, Edie Holmes espouses this view. He says, I've absolutely no problem with people knowing that my views on this issue have evolved as more data have appeared. That's science. Indeed, I've told this to many people. The way I see it is that we set up a hypothesis, then tested it. As far as I can tell, we are only guilty of following the proper scientific method. And he has a clean record here. He was the one who got the pangolin sequence and said 6 out of 6 sites is good enough for me. Second view, here's a quote from Sergey Pond from Temple University. He was not involved in this mess, and from context clues, he was lab leak curious, at least at the time. It started out being a fairly careful discussion with anomalies being aired out and people saying multiple times that there's simply not enough data to resolve this. But at some point, I think there was such strong pressure that they went from let's just wait to get more data to let's publish something that has a very strong opinion favoring one explanation over another without acquiring any new data. I respect both of these viewpoints. I'm not sure that they're not both correct. Revealed in more FOIA emails is some petty drama about who got to be listed as author on the publication and accusations about who stole whose ideas, which is precisely what scientists feud about. But interestingly, this side nugget of information suggests that Fauci's call was dominated by 2 scientists who vehemently argued that the virus almost certainly had a natural origin. The first was Ron Faucier from Erasmus in the Netherlands. We don't know what was said on the call, but Faucier followed up in a very long email on February 2nd, and he detailed his technical reasoning, and then he concluded with, "It is my opinion that a non-natural origin of 2019 nCoV is highly unlikely at present. Any conspiracy theory can be approached with factual information. An accusation that nCoV 2019 might have been engineered and released into the environment by humans, accidental or intentional, would need to be supported by strong data beyond a reasonable doubt. It is good that this possibility was discussed with a team of experts. However, further debate about such accusations would unnecessarily distract top researchers from their active duties and do unnecessary harm to science in general and science in China in particular. The second very pro-natural origin scientist was Christian Drosten from the German Center for Infection Research, though he appears to have given all of his feedback verbally on the call and never returned to the discussion. Later on February 2nd, so that same day, in the follow-up emails, Collins said, "Though the arguments from Ron Fauci and Christian Drosten are presented with more force than necessary, I am coming around to a view that natural origin is more likely." Now, I used this same quote earlier, but I cut off the first part, as is often seen in journalistic reporting on the subject, because it makes Collins look worse without that first part. And ultimately, it seems that they are actually aligning scientifically on natural origin, but there's this pressure to do something. They cannot just let people live. For example, Fauci says, "I agree that we cannot take Ron's suggestion about waiting. Like all of us, I do not know how this evolved, but given the concerns of so many people and the threat of further distortions on social media, it is essential that we move quickly." Why are Collins and Fauci so obsessed with voices of conspiracy? Context. This preprint came online January 31st of 2020. This is the day before the Zoom call. Uncanny similarity of unique inserts in the 2019 nCoV spike protein to HIV-1, GP120, and GAG. A study from a group in India. Preprints are not peer-reviewed yet. During the COVID need for speed, it became fashionable to publish your work online so everyone can see your data and your message while you wait months for that pesky, tedious peer review. This has the unintended consequence of, a lot of things make it through to the public discourse that would have died in peer review. If you ever hear someone say their paper is in peer review or in the process of peer review, red flag that in your mind. This is like eggs that say they are farm fresh. This is like a product that says it is FDA registered. It means almost nothing. And this preprint on HIV inserts rocks scientific Twitter because, number one, the accusation is super inflammatory, and number two, it's completely wrong. 3.5 of the 4 alleged inserts are clearly present in related bat coronaviruses. If it's present in the reservoir, it can't be the result of novel engineering by humans. 2 of the alleged inserts are not inserts at all, but alignment mistakes where the authors just don't even have the sequence lined up correctly to be able to see that the sequences are clearly present in related bat coronaviruses and not insertions. But the last half of the 4th claimed insert is the PRRA that makes up the furin cleavage site, which, to be fair, is suspicious to the other scientists as well. And calling attention to this area of the genome is almost certainly the reason that Christian Anderson noticed the furin cleavage site to begin with on that day and contacted Anthony Fauci. How could this happen? These seemingly coincidental sequence similarities between HIV and coronaviruses, do they mix at some point in history? No. The sequences are far too short for the similarities to be meaningful. If you were to keyword search a document for passive and you came up with pacify and Pacific, we can all understand that those words are related. If you search AT, like at, and come up with cat, attention, disproportionate, and extraterrestrial, we know that those words are not related. The sequence that you queried, AT, is too short to uncover meaningful similarities. That is the scientific community's strong and immediate critique of this paper, along with the inserts not being inserts at all, but alignment mistakes. The authors retracted the paper almost immediately, but it was splashy. Media got wind of it, the Twitter mob latched on. Here's one that I find particularly disheartening. The clever scientisty people have not found any such nCoV spike protein inserts in any naturally occurring virus. It's a mystery, I tell you, an evolutionary mystery. I mean, that's clearly false. They are present in naturally occurring viruses. Senator Roger Marshall, MD, the guy in charge of the Muddy Water Senate report on the origins of COVID-19, he's a physician, he's an educated man, and he is quoted saying, "Additionally, you had a segment called a furin cleavage site that had another spot. It looks like they sliced it out of the HIV virus and put it right to that furin cleavage site. It's just too perfect of a virus." When did he say this, you might ask? He said this in April of 2023. Now, I do not agree with censorship. I'm more of the Ron Fauci attitude that any conspiracy theory can be approached with factual information, and I hate this place we've come to with this misinformation, disinformation language. Big tech makes this up so bad. Nobody trusts anything anymore. Censorship is wrong. But that doesn't mean that bad ideas do no harm. Like, bad ideas do harm. The proof is in the pudding. Scientists in India did something stupid, and it's haunting the Congress of the United States 3 years later. This whole affair really freaked out people like Dr. Collins and Dr. Fauci. The preprint came online the day before their Zoom call. Of course, it colored everything. According to Edie Holmes, there was even some initial confusion on the call that the HIV origin theory was being taken seriously. Everyone was horrified by the direction that things seemed to be taking. The pressure then to do something becomes the root of the problem. They gather more and more evidence leading natural origin, but it's far from absolute, and they need to act like it is. I think some people have a clean record, like Edie Holmes. The penguin sequences were good enough for his conviction. But then Christian Anderson is exhibiting this duplicitous nature where, in an external email on February 4th, he calls engineering a crackpot theory, where to the paper team on February 8th, he says the scientific evidence isn't conclusive enough yet to have high confidence in any of the theories considered, and he's not in favor of publication at that time. And this is cut out of the quote a lot, but he was aware of the pangolin sequences on the day that he wrote this. So again, is there wrongdoing here? It's not an overt cut and dry cover-up. No. Fauci was made aware that the NIH funded Chinese-backed coronavirus research on January 27th. When Christian Anderson called him on January 31st, that information was between two people. If the goal was cover-up, it would be absurd to share this information with 11 other people within 24 hours, including multiple international actors that are going to be less beholden to U.S. funding. I think it is reasonable to conclude that concern was raised. Fauci really did plan to validate the concern and then alert the FBI. But he didn't get validation. He got strong pushback from some and uncertainty from others, then robust discussion over the following days. And at that moment in time, the fear of conspiracy theories causing public distrust and political chaos massively overshadowed any thinking ahead fear that maybe one day, looking back, this was the start of a culture of censorship that ironically seeded massive public distrust. All they were thinking at the time is that conspiracy theories are running amok. They're 85% sure it's natural. They have to do something. I do not know what changed Christian Anderson's mind from being vocally not in favor of publication to being first author. Is it the promise of a grant for his lab? There's not evidence for that, but I'm not sure that there would be. That sounds like a phone call, not an email. But bribery doesn't make sense because they don't need him. No offense to Christian Anderson, but like if Fauci wanted that paper written, it's not like his name is so big that it gives this paper gravitas that it didn't have before. Somebody would have been willing to write it. So I don't know. I don't know what changed his mind. I will never know what changed his mind. So that was Proximal Origin. 3 days after Proximal Origin preprint went online, there was an infamous letter published in The Lancet, which is another prestigious journal. This letter is short, it's like 2 paragraphs, and there's nothing scientific about it. It just says scientists overwhelmingly agree that the virus has a natural origin, and we stand together to denounce conspiracy theories. A number of signers are quoted saying the HIV inserts preprint was a motivating force in their decision to sign. But the organizer of this letter had a conflict of interest that he did not disclose. Peter Daszak organized the letter, and his organization, EcoHealth Alliance, collaborated with Zhang Lixi's lab at the Wuhan Institute of Virology. EcoHealth Alliance is a nonprofit based in Manhattan, and they partner with international researchers and teach them how to safely interact with wildlife like bats to collect samples. And their goal is to prevent spillovers of horrible diseases, and they do so in a scientific way, but also in a policy way, meaning part of their mission is to minimize deforestation and limit the wildlife trade because those are the 2 huge risk factors in virus spillover. So Daszak considers himself an expert in a holistic sense, in a virology plus human behavior and policy. EcoHealth Alliance got a grant from the NIH in 2014 on which Zhang Lixi's lab is a subcontractor with her bat studies in China. But they do a lot of similar work in other places. Studying Nipah virus in bats in Bangladesh seems to be a big one, for instance. Science is a global community. There's not anything out of the ordinary with this arrangement. Here's some details on the grant. It's an R01. An R01 is the standard size of NIH or NIAID funding for a small to medium-sized lab. It's the run-of-the-mill workhorse grant for most scientific labs in the U.S. This grant is for $3 million over 5 years, and about $600,000 or $150,000-ish a year goes to the bat coronavirus research at the WIV. For scale, the NIH budget can fund 17,000 grants this size every single year, so this is not a large amount of money, especially the part that goes to Zhang Lixi's lab itself. But if the NIH only sent $2 to fund Chinese bat virus studies, I think it's not unreasonable to postulate that there might be some concern about optics given the climate at the time. Generally, a conflict of interest has to do with money. Before I present this data, I have to tell you that biotech XYZ pays me as a consultant. I have seen academic researchers proceed to trash the company paying them, which is pretty entertaining. But this idea that you can have a reputational conflict of interest when publishing a letter with no data, maybe you can excuse Daszak for not thinking in that way. But wait, no you can't, because he discussed precisely putting it out in a way that doesn't link back to our collaboration, so we maximize an independent voice. Other involved collaborators pushed back on signing this letter because they feared it would work out in a counterproductive way, or it would look self-serving and we lose impact. So this idea that this is not a good idea was brought to Daszak's attention. He cannot be presumed to be ignorant, and it just does not paint him in a good light. Did Fauci and the Proximal Origin team have anything to do with this letter? Christian Andersen reviewed it on February 4th, and Jeremy Farrar signed it. Both of these people were very involved in the discussions with Fauci, and I therefore think it's unreasonable to believe that Fauci did not know of the existence of this letter. But Fauci and Collins are unlikely to have puppeteered this letter, and the simple reason for that is that the signers are mostly Peter Daszak's network. It makes perfect sense that when finding people to sign a letter, he started with the obvious, the people that he knows. This headline is not totally fair. You can connect anyone with anyone else if you try hard enough. I recognize at least 4 heavy-hitting virologist names that I do not think give a hoot about protecting anyone. I mean, the guy who geniusly thought to test animals in the market during the first SARS outbreak is on here. Obviously, he's going to think the exact same thing happened again. There's no reason to make up that something new happened this time. But there are a lot of names here that I don't recognize, and when I Google them, they're clearly just closely associated with EcoHealth Alliance. Journalists and internet sleuths uncover about 3 additional scandals, all centering on Peter Daszak, though whether he's really at fault or just the victim of sensationalism and political theater is in the eye of the beholder. It's discovered that he was on the ground in Wuhan very early in the pandemic on the WHO's investigative team. I will cover this in my 3rd episode on the market, but the WHO report on the origin of COVID-19 proposed some explanations that were clearly illogical, clearly meant to appease the Chinese side of the investigation team, and it raised eyebrows all over the world. The report coming online was a distinct turning point for a lot of scientists who had previously been holding assumptions of integrity. So Peter Daszak being centrally involved in this scandal is just adding fuel to the fire. The 3rd issue scandal, if we're counting, is that after Fauci's FOID emails were released, it was discovered that Peter Daszak appeared to know Fauci personally. People like Rand Paul were very critical. He said when they call for an investigation, who do they put in charge of it but this Peter Daszak, who's buddies with Fauci, who's thanking him privately in emails for covering up the Wuhan leak. It's all so incestuous. Rand Paul also goes on to accuse Fauci of using U.S. tax dollars to fund gain-of-function research at the Wuhan Institute of Virology via Peter Daszak's company, EcoHealth Alliance. This is the email that drives the accusation that Daszak and Fauci are buddies and incestuous. Tony, CCing David so that you might pass this on to Tony once he has a spare second. As PI of the R01 grant publicly targeted by Fox News reporters at the presidential press briefing last night, I just want to say a personal thank you on behalf of our staff and collaborators for publicly standing up and stating that the scientific evidence supports a natural origin for COVID-19 from bat to human spillover, not a lab release from the Wuhan Institute of Virology. From my perspective, your comments are brave, and coming from your trusted voice will help dispel myths being spun around the virus's origins. Okay. Of the Peter Daszak scandals, this one is the least fair. I don't know what's redacted, but I read this as evidence that Daszak does not know Fauci. First, he CCs Fauci's assistant because he thinks Fauci is going to ignore or miss his email. The whole thing reeks of obsequious subservience. He introduced himself. He introduced himself as the PI of the R01 grant. And he says, "Your comments are very brave." This is not how I talk to my buddies. I don't know if that's how you talk to your buddies. Fauci gives him a short and meaningless reply. Do they have mutual connections? Yes. But to use this as the evidence that these two know each other, that is not a journalist or politician that's acting with integrity, in my opinion. There's a lot more meat on the gain of function accusations. But unfortunately, to understand the context of that hot water, it is critical to understand gain of function and its history. So, buckle up. In 2011, two scientific groups published that they had made a dangerous strain of avian flu capable of transmitting through the air. One group did it by passaging the virus in ferrets, which have a human-like respiratory system. Another did it by making chimeric viruses and also infecting ferrets. But the passaging is easier to explain. They keep collecting virus, reinfecting other ferrets, moving the ferrets further and further apart until they have a virus fully capable of infecting a naive ferret some distance away from the sick ferret. So they're using evolution to their advantage to select for mutations that allow for aerosol transmission. Why were they doing this? The purported purpose is to say, hey, that avian flu that people keep getting from birds at markets, that virus with its 50% death rate, that virus could one day be passed efficiently between humans. We need to take avian flu seriously. The scientists had permits, funding, planning, permission, oversight to do this work. They took precautions. They had special facilities. They had vaccinated the lab workers for H5N1. But when these papers were published, there was collective outcry from scientists, the public, the government who allowed the work in the first place, because they intentionally drove a very dangerous human pathogen to go airborne. This kicked off a years-long chain of events and divided scientists between those that felt the knowledge gained outweighed the risk of this type of research, and in this camp are Fauci and Collins, and those that believe that this work should have never been done, and they start organizing and advocating for safer science. This is by no means an exhaustive list, just some examples of outspoken individuals here. Eventually, this advocacy led to government regulation, and when it comes to defining gain of function in legislation, everything quickly becomes very muddy because all of virology research could be viewed as gain of function in the right lens, but something is clearly different with the flu experiments. If I were the queen and were to define it, I would say there's two elements. There's one bucket, what are you working with, and another bucket, what are you doing? In the what are you working with bucket, there are select agents, human pathogens, and things not known to make humans sick. Select agents are the scariest: smallpox, Nipah, Hendra, SARS, dangerous avian flus. Ones that are airborne would clearly be of the most concern. Human pathogens are like anything that makes people sick, but not the scary select agents. And then there would be, stuff that's not known to make people sick, like AAV, foamy viruses. There's literally a gazillion. In the what you're doing bucket, experiments on proteins and pseudoviruses, this is unquestionably the safest. The whole virus is not there, or at least not there in a form capable of replicating. So that's pretty safe. The next step up in danger is making chimeric viruses. That's swapping genes or parts of genes to narrow down the parts that are important. These type of experiments answer what is necessary, what is both necessary and sufficient for XYZ function. Then in the real danger zone would come things like bioengineering an element that would be expected to increase transmissibility or pathogenicity, or passaging or combining passaging with any of these other strategies. We really don't want to harness the power of evolution to make a dangerous virus more dangerous. As a reasonable person in this hypothetical world where I'm queen, I think I would define research of concern as maybe these activities in select agents, especially those that are respiratory. But all this to illustrate the complexities in definition stack up quickly. This is not conducive to legislation. October 2014 is when the gain of function research moratorium began. The White House and the Department of Health and Human Services, they ended up defining gain-of-function research very narrowly as work on influenza, SARS, or MERS viruses only, and work that would confer attributes reasonably expected to enhance the pathogenicity or transmissibility in mammals via the respiratory route. The funding moratorium was in place for a little over 3 years, where it was dropped in lieu of a committee review process. The committee review process re-allowed funding and performance of any gain-of-function research as long as the committee decided that the benefits outweighed the risk. So starting in late 2017, the committee literature states it's concerned with potentially pandemic pathogens, which are defined as 1, likely highly transmissible, and 2, likely uncontrollable spread in humans, and 3, likely highly virulent. It specifically excludes pathogens circulating in nature like bat coronaviruses, which according to Peter Jajak represent a clear and present danger to humanity. To reiterate, as of December 2017, the committee reviewing gain-of-function research specifically did not want to be bothered with reviewing bat coronavirus experiments. So under both governance scenarios, the agency put a very high bar on what qualified as concerning, and they erred on the side of regulating almost nothing. In retrospect, that does not play well. But one way to think about why these rules seem so lenient is that they were meant to stop people from deliberately making viruses more dangerous. They were not written to stop people from accidentally making viruses more dangerous. Attempting to identify what features of a spike protein enable a jump to humans, there's a very different spirit to that experiment than deliberately making a dangerous influenza go airborne. This is how Anthony Fauci can so confidently say over and over that the NIH did not fund gain-of-function research in Wuhan. Anthony Fauci's definition of gain-of-function is work reasonably expected to enhance the danger of SARS, MERS, or flu viruses only. Rand Paul's definition of gain-of-function is souped-up superviruses where they put a protein in the spike protein. And I'm not belittling Rand Paul's scientific illiteracy. Putting a spike protein in, or like putting a protein in the spike protein, this is not language that is quite right, but I get what he's trying to say. In their infamous fight on C-SPAN, inexplicably, neither party acknowledges the obvious, which is that this is not an argument about what the NIH did or did not fund. It's an argument about what should qualify as gain of function. Rand Paul is not in the right. Fauci did not lie before Congress. He does not deserve to go to jail over this. The agency enforced the rules as written, and yet the public doesn't seem cool with it, so I think we have a problem. That's the angle that Rand Paul should take. Why are these rules so lenient? You wrote them. What gives? Despite very few projects falling into the definition of gain of function during the moratorium, the EcoHealth Alliance WIV collaboration did raise some flags. The R01 grant was issued in 2014, prior to the moratorium, but the work was flagged in a 2016 progress report. Specifically, a proposal to make chimeric viruses with the MERS virus and also the SARS-like backbones that I described before. The series of email negotiations is what people refer to when they say Dajak was sidestepping gain-of-function rules. The scientists have the same goal as always. They want to test novel spike proteins by swapping the new spike into their favorite backbones and infecting cells in mice. If the mice get sick, the spike joins this elite class of super scary spike proteins that we can compare and contrast and understand more about what risks lie in the reservoir. After discussion with the agency, it's decided that the experiments do not fall under the definition of gain of function because the SARS-like viruses are bat coronaviruses, not SARS itself, which seems like a loophole, but importantly, the proposed chimeras are not anticipated to confer enhanced pathogenicity. This is the argument. There's an inherent pathogenicity and transmissibility of MERS. By swapping in a novel spike protein, the best that you can reasonably expect is that the novel spike protein will work about as well as the native MERS spike, which already infects human cells no problem. It will probably not work as well. This is what my entire virology career is screaming at me. It will probably not work as well. Therefore, if you're already taking all precautions you would with regular MERS, which is terrifying and dangerous, that should be sufficient, in theory. I completely understand the rage as all of these pieces of information trickle out. The public clearly thinks that making chimeric viruses is a needlessly dangerous activity dreamed up by soulless dystopian mad scientists. And like, point taken, maybe the benefits of that type of research do not outweigh the risks for the more dangerous human pathogens. But what happened in the past, the rules as written were very lenient, and the people writing the rules did not even think through all the possible scenarios, so they were interpreting the rules on the fly. But importantly, by 2018, according to a leaked proposal, Zhang Lixi's lab had spent 14 years looking for bat coronaviruses, and they had only uncovered 3 that they could use as a backbone in a reverse genetic system in addition to regular SARS and regular MERS. SARS-CoV-2 is not any of those viruses with a novel spike protein. It's far too divergent. Again, what we would expect if COVID came from the controversial experiments is that it would match one of these backbones with a completely novel to the world spike protein, and it doesn't. There's way too many mutations, too evenly sprinkled and spread out throughout the genome between COVID and her favorite 3 SARS-like viruses. I didn't include MERS on the slide because MERS is so different that it's hard to even align and be interpretable. But again, it would be very, very obvious if COVID came from any of the experiments described in the EcoHealth Alliance grant, or any of the updates, or any of these leaked emails. The controversial gain-of-function work is not the origin of COVID-19. These experiments, risky or not, wise or not, illegal or not, reported on time or not, they're not the origin. Is this discussion a good ethical sanity check? Yes. But this is not evidence that COVID came from a lab. But frustratingly, many people think that the fact that they were doing gain-of-function work is strong evidence that COVID came from the lab, but it's not. Is it possible that the group was conducting similar undescribed work? Absolutely. I'm just trying to point out that understanding precisely the work that was proposed and negotiated in private emails years ago makes the possibility look more remote, not more probable. The window of plausibility is closing, not opening, because that's a hell of a needle to thread, to have the one thing that escapes your lab be something never published, supposed to do, never mentioned in any email when the emails were assumed to be private. Everything I've talked about thus far pertains to the early events, January through March of 2020. During the latter half of 2020 and certainly through 2021, the scientific community's commitment to natural origin really started to wane. In my opinion, this is mostly due to learning that Zhang Lixi's lab would do the initial handling of bat samples in BSL-2. In retrospect, the heebie-jeebie factor is high. Working with bat blood in BSL-2 is technically sufficient, even by current standards, but maybe that feels wrong. Maybe we need to rethink that. We got this information from Zhang Lixi herself in a response to questions posed by Science Magazine. Her answers are obviously going to be heavily censored by the Chinese government, but she does describe the work conducted under the BSL-2 and the BSL-3 conditions, and she's probably trying to push back against the idea that the virus came from her BSL-4, because the BSL-4 is a more central part of the lab leak theory in China. There was local pushback when it was built. BSL-4 was one of the first phrases censored on WeChat, right alongside Huanan Market. But in the West, we don't have this clear attachment to which building the leak came from. These pictures of people in bubble suits insisting that that's not where they were doing the coronavirus work, like, that's not actually comforting that they're doing work in a less stringent environment than that. Likely because of the safety concerns, at least 3 signers of the Lancet letter walked back, and here are quotes from 2 of them. I believe that a thorough investigation about the origin of COVID-19 is needed. A lot of disturbing information has surfaced since the Lancet letter I signed, so I want to see answers covering all questions. Another, I'm convinced that what happened is that the virus was brought to a lab, they started working with it, and some sloppy individual brought it out. They can't admit they did something so stupid. Other famous scientists were starting to speak out too. David Baltimore is a Nobel Prize recipient for his work on retroviruses. He said, "When I first saw the furin cleavage site in the viral sequence with its arginine codons, I said to my wife it was the smoking gun for the origin of the virus. These features make a powerful challenge to the idea of a natural origin for SARS-2. Ian Lipkin says, That's screwed up. It shouldn't have happened. People should not be looking at bat viruses in BSL-2 labs. My view has changed. Now, the other part of this quote, which is often left out, is that's still not direct evidence of a lab leak. There is no proof of a leak. But then Robert Redfield, head of the CDC in 2020, still believes today that COVID-19 is the result of a lab leak. His reason for believing this is that the virus replicates too well in human cells for it not to be the product of design or serial passage. As frustration grew, a group of scientists wrote a letter to Science magazine in May of 2021. They said there isn't enough evidence to disprove lab leak. They were not convinced by proximal origin, not enough to say it's a done deal the way that everyone is acting like it is. All of this crazy information has come out in the past year about safety standards in the Chinese labs. The WHO report is suspect. They wrote, "We must take hypotheses about both natural and laboratory spillover seriously until we have sufficient data. A proper investigation should be transparent, objective, data-driven, inclusive of broad expertise, subject to independent oversight, and responsibly managed to minimize the impacts of conflict of interest." That sounds great. That's all we want. Several signers of this letter remained central figures in the lab leak debate over the next few years. Jesse Bloom is the first author, the John Hancock. He goes on to be this perpetual thorn in the side of other scientists trying to demonstrate a closed case for natural origin. He's clearly upset about transparency, and he picks a big fight directly with Anthony Fauci. I'm going to talk about Jesse's work in the episode on the Wuhan market. Michael Warraby is a self-described lover of a good conspiracy theory, but he goes on to generate some of the strongest data to support natural origin and becomes an advocate for natural origin. And again, this will be covered in the Wuhan market episode. Alina Chan was at this time a postdoc at the MIT Broad Institute, so she's early career, and she's one of only a handful of scientists who actually lean lab leak as opposed to natural or neutral. And by taking a stronger stance than others are willing to, Alina Chan gets a lot of attention. She's charismatic, she's well-spoken, she does interviews with journalists. She's interviewed by Sam Harris, she publishes opinion articles in Medium, and she gets a book deal, and she publishes Viral. And she becomes this Twitter darling for lab leak hypothesis. Despite her media presence, Alina Chan only has 2 scientific papers on the topic of origin. The first is a preprint from May 2020. The second is on the Fear and Cleavage site. The first paper was pretty early on in the pandemic, and notably, this work has not been published in a journal in the 4 years between then and now. This is the money figure from the preprint, and I understand that these trees are not super approachable, so hang on to your seats. Trees like this one show the genetic relationship of one sequence to another. So each dot is an existing viral sequence, and an algorithm then decides how closely related they are to each other, very much like a family tree. But on this one in particular, longer branches means more mutations. And Alina Chan's big point here, and her first point in her Sam Harrison interview, and a point heavily emphasized in her book, is that the first SARS in 2003, upon jumping to humans, had to acquire a lot of mutations in rapid succession during initial adaptation to a new host. In a new host, maybe there are a lot of things that aren't quite right, and the virus is under selective pressure to change rapidly. And then once it gets good at replication in humans, it can kind of cool its jets. So in the first few months, you see all of these nucleotide substitutions. Then later in the epidemic, the mutation rate is more stable. And she argues that this later phase, when the virus is good at replication in humans, looks a lot like SARS-CoV-2 did right at the beginning. There was no period of rapid mutation acquisition. It was pre-adapted to humans somehow. I wonder how. Stay tuned. The last signer of interest in this story, at least, is Ralph Baric. If they're paying attention, most people know Ralph Baric from the DARPA proposal, the metaphorical giant other shoe dropping in this saga. DARPA is Defense Advanced Research Projects Agency, and Peter Daszak, Zhengli Shi, Duke Professor Lingfo Wang, and UNC Professor Ralph Baric, and then also some lesserly involved characters, submitted a proposal to DARPA in 2018 that was rejected. In September of 2021, it was leaked by The Intercept. One paragraph is of extreme interest, and are you ready for this jelly? They proposed inserting proteolytic cleavage sites into coronavirus S genes. They proposed the work that I described before, where they're going to gather a bunch of bat sequences, test the spike proteins for ACE2 binding, but then they're going to analyze these spike sequences for appropriately conserved general proteolytic cleavage sites and for potential furin cleavage sites. Where clear mismatches occur, they will introduce human-specific cleavage sites, end quote. Then they will put the winners into their WIV16 and SHCO14 backbone, evaluate growth on cells, see if spikes in these new backbones have the potential to make mice sick. The coincidence is stunning. We have all of this hand-wringing about this potentially engineered furin cleavage site, and then it comes out that the lab proposed to do something fairly similar in 2018, which is the year before 2019. Spooky. Ralph Barrick is the collaborator on the DARPA proposal that would have been engineering the proteolytic cleavage sites and hypothetically would have been performing this work had it been funded, though that's debated, unclear. Ralph Barrick has historically been this careful proponent of gain-of-function research because there are some specific applications where making and testing chimeras really is required for vaccine development, and the moratorium really affected his projects. Interestingly, Ralph Barrick signed the letter saying investigate the origins of COVID-19 in May of 2021. So that's prior to the DARPA proposal leak in September of 2021. And I'm not sure of his motivations for signing the letter, except that he appeared to take issue with the WHO's immediate conclusion that the lab leak was extremely unlikely. He said, that conclusion is premature given the lack of conclusive data and China's more relaxed laboratory standards. He also said, the origin of the virus that started this pandemic is worth discussing, worth investigating, but conspiracy theories that have no foundation in science do nothing but cause harm and stress and damage to people like myself. Why is Ralph Barrick being harmed? He is the target of a particularly large amount of vitriol online because he's blamed for engineering that furin cleavage site and creating the pandemic. He's also accused of knowing Fauci, but Fauci not knowing him. He wrote a comment in the DARPA proposal instructing Zheng Liqi to downplay the specifics of the location of the work because the agency would, quote, freak out. He's accused of offshoring experiments that he would have done locally in the absence of the funding moratorium and having Zheng Liqi's lab do them instead. He also, his work assisted in development of the Moderna vaccine, so that's the seventh deadly sin. Here are some mean quotes on the topic. You and your maniacal flunkies caused the murders of 1 million Americans and 7 million people globally. A reckoning is coming. Enjoy your freedom while it lasts. Here's another. Barrick Lab made the virus. He also made the cure. Fauci's buddy, Dr. Ralph Barrick, created COVID in the UNC Chapel Hill lab, and U.S. taxpayers paid to transfer his work to the Wuhan lab. Science Magazine sponsored a debate with this cast of characters, Alina Chan, Jesse Bloom, Lingfa Wang, and Michael Warabi. Despite the interesting scientific discussion, the main takeaway for most viewers is probably that Alina Chan really drifted off the course of scientific topics to lay into Lingfa Wang and pin him, simply repeating whose idea was it, whose idea was it, whose idea was it to engineer the furin cleavage site. And of course, Lingfa Wang says, Barrick's. Of course I knew. So unfortunately, Ralph Barrick needs to have security outside of his lab and home at all times. As time went on, though, people noticed that features of the furin cleavage site that were not consistent with intelligent design. First of all, there's this extra amino acid. This proline never made sense. It's part of the insertion, but it's not part of the furin cleavage site. Secondly, the sequence is RRAR. It would work a lot better if the sequence was RARR. Why would anyone insert a suboptimal site? The third quality that is strange is codon usage. There's 20 amino acids coded for by 3 RNA letter codes or codons. But there are more 3-letter combinations than there are 20, so there are multiple ways to code for each amino acid. So for example, there's 6 ways to code for arginine: CGU, CGC, CGA, CGG, AGA, and AGG. Organisms have codon preferences, which can be strong, And suboptimal codons can have profound consequences for replication. Not codon optimizing your sequence is total amateur hour in molecular biology labs. It's the type of thing that you do once in grad school and then you never make that mistake again. You always run your sequence through a codon optimization algorithm. These CGG codons are rare in coronaviruses. They are the least favorite of the 6 ways to code for arginine. This is not how you would design it. But here's the bombshell you've probably never heard of. In late 2021, a French group from the Institut Pasteur found the closest bat virus to SARS-CoV-2 that had ever been found at the time. It's not the immediate progenitor by any means. It only just barely beats out RATG13 from Zhang Lixi's lab at 96.8% total genome nucleotide sequence identity. So that's still about 30 years of evolution away. And the French scientists found this sequence in northern Laos, about 1,000 kilometers from the caves in Yunnan where Zhang Lixi likes to sample. They name it Bainal 2052, and importantly, it has a nearly identical receptor binding domain to SARS-CoV-2. It differs by only 1 amino acid in this whole thing, and just a handful of silent point mutations. And 6 of 6 key ACE2 binding sites are identical. That's compared to 1 of 6 in RATG13. This observation obsoletes any shadiness with the pangolin sequences. Who cares what they were trying to accomplish? We have a much better sequence right here in the bats. But does it have the furin cleavage site? Bainal 2052 disappointingly does not have the furin cleavage site. The group collected and sequenced a handful of samples from the same cave that are also very close to SARS-CoV-2, and they published these sequences as well in that same paper. They just make a big deal that Bainal 2052 is the most similar, but there's clearly similar strains floating around in the cave. Let's look closely at my alignment. The algorithm is confused. It has split this proline codon, and that's definitely not right. The algorithm is going off of the nucleic acid sequence and trying to line them up, but of course a split codon indicates that something is off. Also, this arginine looks pretty important. It's widely conserved, far beyond this list of closely related viruses, and I want to make sure that those R's line up. And look how poorly this part matches at the nucleotide level. This is just, something's wrong here. What I'm seeing is NSP, and then NSP in Wuhan 1. What happens if I fix this? So I move NSP over to line up with NSP, and then I move arginine over to line up with all these other R's. Well, I'll be darned. That looks a whole lot closer to something that could turn into a furin cleavage site. Look at this serine codon. It's UCA for all of these sequences, and then in Wuhan 1, it's UCU. That third wobble spot is different. Look at the alanine GCG codon for the sequences at the bottom. It's GCA in Wuhan 1. That third wobble position is different again. It's a crossover. It's an out-of-frame insertion. There was one bat that had multiple strains, and they mixed. Zhang Lixi reports that 6% of bats are positive for infection at any given time during the flu season for them, which seems like a small number, 6%, but if you got 40 million bats, that's a lot of chances for co-infection. Natural chimeras, where the viruses kind of reshuffle their genomes, these are very common. Every coronavirus is chimeras of many others. They're reshuffling their genomes all the time. So this old UCA serine codon is split. Here's the replicase, chugging along on a sequence that looks like Bainal 2052. Replicating my genome. Beep, beep. Oops, I fell off. Oops, I got lost. Oh, here's another strand. Okay, back home. By pushing these two alanines upstream by the CA back half of that split serine codon, an alanine is maintained at this site, and it becomes the GCA alanine codon seen in Wuhan 1 instead of the GCG of these other strains' alanines. At that point, the sequence is 2 mutations away from making a furin cleavage site amino acid sequence. Stated another way, intelligent design would put an insertion between codons. That feels right and rational and as God intended. Evolution can't see codons in a nucleotide sequence. It's just a strand of RNA. The sequence gets shoved in, it happens to split the serine codon. This is a sequence that's there today in the COVID virus, and this sequence exists at that location in a genome in a strain 30 years of evolution removed. So to me, as a molecular virologist, it is now completely unreasonable to believe that the furin cleavage site is anything other than natural. For you skeptics that think that all data can just be made up and these sequences could be falsified, the original authors did not notice this near furin cleavage site sequence. The word furin appears 17 times in the publication, and it's all in the context of, despite the absence of a furin cleavage site, these sequences still seem really important. They are so disappointed. It is left to other scientists who read the paper to figure it out. I'm not sure who to credit. Michael Warraby, who adorably printed out an alignment to show on the Science Magazine sponsored Zoom debate, or Bob Gary, who published this 3-paragraph rage of a paper that may as well be entitled, "Can Lab Leak Die Already?" Let's go back to Bainal 2052. The receptor binding domain is what we think of as the single greatest contributor to a coronavirus being able to infect human cells. It's the physical interface between the spike protein and human ACE2. There is a bat virus with a nearly identical receptor binding domain to the original COVID strain, and it's not some random virus in the bat reservoir that happens to have an identical binding domain sequence. It's the closest thing to SARS-CoV-2 that anyone had ever found at the time. And it's in the same cave as the strain with the furin cleavage site precursor sequence. So this argument by Alina Chan, and later picked up by Robert Redfield, is not valid. That the virus is suspicious because it didn't have to work as hard getting mutations. That's just not substantiated. We literally know that a crossover between two viruses in the same cave would yield a sequence only 2 mutations away, and that's 30 years of evolution removed. A sequence that looks like SARS-CoV-2 is definitely in the bats. In fact, this is a physically impossible number of mutations for one virus to acquire between January and March of 2003. That's only 3 months. This is more than the mutation rate of beta coronaviruses. A much better explanation for Alina Chan's seemingly disturbing observation is the number of jumps from palm civet to human. The early diversity with SARS in 2003 was likely the result of multiple strains from the market jumping into humans. We know from epidemiology that there were at least 5 independent jumps, but there were probably many more. They had no idea where SARS was coming from, and they let it rip for a while. The later lack of diversity is that most jumps die out, and a few successful strains make it a little further, given the appearance that evolution is slowing down later in the outbreak. But it's not one virus acquiring a lot of mutations, like evolving fast and evolving slow. It's a number of viruses and then fewer viruses. That's what makes it look like there's less evolution and the mutation rate is more stable. So viruses are kind of born on different bases with regards to their molecular landscape or the number of mutations they are away from being successful in a new host. If a virus needs 50 mutations all at once, it's just never going to happen. It has to hit a home run, even less chance than hitting a home run. If it only needs 2, that virus is like born on third base. SARS-CoV-2 did not need bioengineering of a furin cleavage site. It did not need to be serial passaged to acquire the ability to replicate in human cells. It did not need gain-of-function experiments. No matter what the scientists said in the emails, no matter what they named their Slack channel, no matter who lied or did not lie before Congress, the virus did not need human intervention. It was just waiting in the reservoir. Everyone leaning lab leak, by everyone meaning Alina Chan, Rand Paul, Fox News, U.S. government reports, etc., they use pretty much the same pillars of argument. The scientifically suspicious things are the furin cleavage site, the pre-adaptation to humans, and the fact that there were no positive animals identified at the market. The circumstantially suspicious argument is that there was gain-of-function work arguably occurring in Wuhan. The Wuhan location coincidence when lots of coronavirus bat work was conducted there. The emails indicating scientists were leaning lab leak initially. The safety concerns at the WIV. The hot topic origin of RATG13. And the removal of Zhang Lixi's sequence database from public viewing. These bat viruses knock out 4 of the arguments in one fell swoop. The furin cleavage site, which is inextricably tied to the Zoom call and the scientist emails, the pre-adaptation to humans, and the gain-of-function work. It really takes quite a bit of the wind out of the sails of lab leak, because what would have happened if sequences like these were available in early 2020? The furin cleavage site was the entirety of the concern expressed by the scientists on Fauci's call. There would be no explosive FOIA'd emails from the scientific experts, quote-unquote, warning Fauci that it might be engineered. They would not have had a call in the first place. You would not have the well-respected David Baltimore's chilling comments about the furin cleavage site smoking gun. He has since completely retracted that statement, and he is on Team Natural Origin now. The DARPA proposal would not have been leaked. You know the razzle-dazzle, the reason it packs the punch, is that paragraph about inserting proteolytic cleavage sites. And you probably don't have Fauci and Collins tying their own nooses quite so tight and pressuring the publication of proximal origin in response to the fears of rising conspiracy voices. There is likely no proximal origin paper if the furin cleavage site is presumed to be natural. Poor Ralph Barrick. Like, what is his crime? I think the death threats have probably died down by now, but it's all really tragic and senseless. In the beginning, scientists saw 3 options. SARS-CoV-2 was an actively malevolent bioweapon. SARS-CoV-2 is a virus that was engineered or serial passaged and it leaked out of the Wuhan Institute of Virology, or it was a natural zoonosis from the Wuhan market. Nobody but Steve Bannon seemed to think it was a bioweapon. So because it is not these, then it must be this. Remember that the conclusion of proximal origin is our analyses clearly show that SARS-CoV-2 is not a laboratory construct or purposefully manipulated virus. They don't consider at the time the possibility that the virus leaked from the lab, but in an unaltered form than the virus in the bats. Not engineered, no gain of function, just exact same sequence as a bat in a cave. There still remains a possibility that grad students digging through guano in remote caves is as good of a conduit for viral spillover as the wildlife trade. So catch my next video, which is going to be all about the lab.